It's my pleasure to introduce Megan. Megan has been working on her master's degree with us in the distance program in plant breeding. She's doing this while keeping a full-time job with Bayer, previously with Monsanto. And she came to us already having some graduate work from North Dakota State while teaching and doing technical work, and had her undergraduate work at Missouri, I believe. But she was willing to take on the dangerous and illicit topic, as you mentioned, Scott, on working on how to optimize indoor grows operations and inputs, and somewhat automate them for cannabis. And I look forward to her talk. Megan.
All right, thank you. Can everybody hear me and see my screen?
I can hear you great.
Okay, can you see the screen? Okay.
I can see the screen too.
Okay, great. So thank you, Dr. Jessup. Today I'll be going over a project to develop a growth protocol for drug-type cannabis in large-scale commercial operations. Some of the things I'll be going over is some growth, how the cannabis industry has grown in pretty much the last decade, how it's classified currently, some of the challenges when creating a commercial growth protocol, and how I used some modified hemp scales to establish growth stages, and then metrics to evaluate the growth protocol. And then finally, I'll be going over stage by stage the actual growth protocol using a couple of example varieties. So as I said, cannabis has grown in the last decade. For instance, as of 2022, medical cannabis is now legal in 38 states. And as of 2021, globally, it's legal in most of North America, South America, Europe. It's legal in Australia and New Zealand, and is a growing market in Africa. And so it's been legalized for medical and recreational use and has been increasingly recognized for therapeutic and medicinal benefits. And the actual product of drug-type cannabis is the unfertilized flower buds of female plants. And they contain psychoactive or psychotropic chemicals, the two main ones being tetrahydrocannabinol, which is referred to as THC, and cannabidiol, CBD. So there's a picture of the cannabis flower on the left with the buds. You can see they're kind of bright colored. And so the traditional use is smoking the buds, but there are now a variety of other products that have been developed. And I've got an example just from one company of the diversity of products that have been created. So there are vaporizers which release cannabis oil. I think of them like incense. There are lots of inserts for vape pens and things. There's flour-like and distilled cannabis additives for food or to ingest directly. There's pills and capsules, lozenges, which are kind of like hard or chewy candy, and then lotion. And you can actually absorb the active ingredients in cannabis through the skin. Okay, so how is it classified? So it's classified in two different ways: phenotype and chemotype, which is essentially another type of phenotype. So when breeders are looking at cannabis, they're looking at the height, the smell, the number of days to maturity, if a photoperiod treatment is required for the reproductive stage, the leaf type and shape, flower type and shape, flower size, and the effect on the user and the potency. So there are 3 subspecies of cannabis, and I have them in the diagram in the middle, and then live pictures of them. So the one on the left, the tall one, is a sativa or narrow leaf drug type subspecies. They can be up to 20 feet tall and they're narrow. And their leaves also kind of reflect that. They've got long, skinny, light green leaves with long leaf blades. You can see it on the left, long, thin leaf blades, long flower clusters, and they're said to be higher in THC. They originate in warm tropical environments in China, Thailand, and India. So the next subspecies is the plant in the middle of the diagram. So it's an indica or broadleaf drug type. So they're much shorter, as you can see, bushier, broad with dark green leaves. They have small, dense flower clusters and are supposedly higher in CBD. They originate in hot, dry climates like Pakistan and Afghanistan. And then finally, there's ruderalis, which this one's debated, so I don't go over it quite as much, but it's the really small, skinny one there on the right. And it's mainly used for its autoflowering characteristics. So the other two subspecies need a short day to go to reproductive growth. It does not. So it's often hybridized with the other varieties to give them this attribute of where they don't need a photoperiod to be reproductive. And it originates in Russia and the east-west border countries of Russia. And so there's also hybrids of all three of these. So there's lots and lots of different varieties of this plant. So the second classification is chemotype, and this is how the different cannabinoids, so THC and CBD, relate to each other. There are other cannabinoids that contribute to the potency, but THC and CBD are the most commonly referred to. So I'm going to go over the first three since we're doing drug type cannabis. So chemotype 1 is higher THC, lower CBD, and you can see in the diagram there at the top, the little scale, and it is associated with certain effects on the user. So I have a chart to the left which say in blue like what the effects are. So it's more relaxing, more sedating, your mood is likely to change, and then they have recommended medical conditions it may treat. So autism, HIV/AIDS, drug withdrawal, and you could loosely correlate it with sativa. So none of this is confirmed, but this is what it's purported to help with. So chemotype 2, the yellow there, has about equal levels of THC and CBD, and it's moderate on that scale to the left. So moderately relaxing, sedating, moderate mood changes, and can be for fibromyalgia, multiple sclerosis, or mental emotional pain, which is pretty vague. And it's unclear whether it is also associated with sativa types. And then finally, the third chemotype is the higher CBD than THC, and it is more energizing, less relaxing, no sedation, and no mood changes. Could be used for epilepsy, heart disease, and acne. So this brings to some of the challenges with developing a commercial protocol for cannabis, because all of those attributes I just went over are really pretty subjective and they don't correlate well to each other. They're not really based on a ton of research. It's just cultural knowledge that's been passed from person to person. And in fact, they've done some genotype by sequencing studies that have shown that there's inconsistency in the labels. So something that's labeled as 100% sativa is actually grouped with indica varieties genetically. So the labels don't really mean much. And so this creates a challenge for establishing a cultivation protocol. One is that commercial cannabis is a young field and there's not a lot of research out there. The factors to determine the growth conditions are undetermined or really, really specific. So I've read a lot of papers, but they're usually for one variety, one environment. And optimum temp, humidity, fertilizer, they say in the articles, hasn't been determined yet. So it's hard to apply one protocol across all the different varieties. Another challenge is the past illicit growth strategies and breeding. So for example, strains, cultivars, lines, and I use all those interchangeably. They're not easy to delineate to help with growth protocols. I don't know if the variety I think I'm getting is the variety I'm getting. So I'm going to give you an example. So this strain on the bottom right is called Blueberry. So this is actually from a cannabis database. It says that it's a landrace, but it doesn't specify where it's from. It says the product is very different between individuals due to crossbreeding, and that the only way to tell this variety is that it tastes like blueberries. So that's kind of difficult to apply to tell what you're getting in a variety. So for instance, something could be a hybrid with a blueberry variety and express that trait where it tastes like blueberries. So it doesn't really denote how it'll perform. It doesn't tell me where it's from, what environment it has adapted to, et cetera. The other thing about the illegal or illicit growth strategies in breeding is there are wildly different growth strategies that are hard to compare. So they're grown in soil and high tunnels or outdoors. They're grown in basements. This one is grown in a closet. So it's hard to compare these growth strategies, even though there's a lot of information about how they grew them. So I had to basically build this protocol by combining the academic studies with the underground growth methods, and then also cannabis manufacturer recommendations, as well as suggesting what type of space to put each phase of cannabis in. So the first thing I needed to do was break the process down by stage and determine if I'm comparing the same thing in the academic literature as I am in the informal growth protocols. So this is where the BBCH scale comes in. So it's a modified hemp scale, and I have a picture of the general phases of hemp here. And it gives physiological indicators of stages in hemp. There are not well-established stages in cannabis. So I can match a physiological growth indicator to days after planting in informal protocols and kind of figure out commonalities. So out of this, I'll be covering principal growth stage 0, which is germination and sprouting. There's principal growth stage 1, leaf development, the vegetative stages, and the reproductive stages. And then I've added a principal harvest, post-harvest. Sorry, I've added a harvest and post-harvest stage, which wasn't originally in the scale. And I'm actually gonna skip stage 1 because it's often confounded with the vegetative stages. They overlap. So for the sake of clarity, I'm just gonna go right from germination, sprouting to vegetative because they overlap. Okay, and then the second thing I did to establish a growth protocol was I wanted to establish a baseline protocol with the research currently out there, and then to judge how effective that protocol is, use it on tester varieties and collect metrics. So that's what I'm showing here is examples of some of the metrics you collect. So I establish a growth protocol based on what I know from the research, and then I use indicators to determine how effective my protocol is for a test variety, is the idea I'm going for. So one of the categories is a metric on plant stage. So again, that's matching that days after planting to physiological growth stages on that hemp scale. So how many days after planting does the vegetative stage begin and end? And how do I know that based on plant physiology? The second bucket are environmental metrics. So what is my growth environment look like? When are the plants given irrigation and fertilizer type and rate, the temperature, humidity, and lighting settings? Those all help me establish the baseline at which I'm growing from. Another category is production metrics. So what are the different characteristics of my production system? What are the variables that I factor into my plant growth? So what's the variety I'm using to test with? Is it a seedling or clone? What's the flattened pot sizes I'm using? What's the media type, the watering method, etc.? And then those are all combined to then judge these plant performance metrics to see how effective my growth protocol is. So that would be things like germination and emergence percentage, the number of stem nodes and when they occur, chlorophyll readings, phenotypes like wilting or chlorosis. So all of these things are meant to help take a variety, which is an unknown with unknown growth conditions, and determine how well a protocol is working, and then allow me to factor in variables to tailor that protocol to a particular tester variety. Okay. So I will be using 2 example tester varieties. One of them is Eldorado OG, which is referred to as an indica variety. I have pictures of the plant in flower, a harvested stalk, and the seed. And then the second variety is Original White Widow on the right there. So I have a plant in flower, a close-up of the flower stalk, and then the seed, and it's considered an indica sativa hybrid, although sources differ on that. So it kind of shows the ambiguity there. Just real quickly, There are two types of cannabis production. There's clonal propagation and seed-grown cannabis. Clonal propagation is taking propagules from a mother plant, which are identical to that mother plant and identical to each other. And then seed-grown plantings can be used to develop or maintain a cultivar or inbred. And because cannabis is dioecious, it means that there are separate male and female plants. So when growing seed, half will be male and half will be female. This is good for breeding programs, but it makes a highly variable growth protocol if you're just producing. However, I'm going to go over the seed-grown protocol and not the cloning just for sake of time. So getting into the actual protocol, pre-planting, so we're in stage 0, germination, sprouting, and emergence. And pre-planting, I want to look and inspect the seed. So I want to make sure that I'm planting dark, hard-shelled seed. That indicates it's mature. And the green, soft seed is immature. And I want seed in the middle size range because experiments on hemp have shown that that has the highest germination percentage when tested. And so there's the seed on the bottom two pictures are the seed for our example varieties. So day 0, I'm inspecting the seed and then seeding. So you can seed into flats is what I chose for my protocol. And you can see there's a variety there of different types of flats that you can grow cannabis in, planting about 1/4 of an inch deep. And then for the example varieties, and I got this out of grow guides from different sources, largely the seed distributors. Both of my example varieties emerged on day 3, and then their cotyledons were fully unfolded on day 7, which marks the end of the germination sprouting stage. But basically you need to plant seed in a sterile media that allows air, water, and nutrient exchange, and field soil is not recommended. So as far as spaces go, the first room I would suggest for a commercial cannabis operation is a seedling or clone room. So seedlings are kept at different environmental conditions. They're usually lower light, lower nutrient, higher humidity and temp than vegetative or reproductive plants. And there are several manufacturers like Conviron, for example, that market to cannabis growers. So I've got a picture of a Conviron seedling chamber there. So key elements in this space are an automated environmental control system, which can monitor and program environmental conditions and watering, irrigation, fertigation. This one has a hydroponic irrigation system with ebb flood benches. So basically it's a bench that you put flats in. You can see in the diagram on the right. Water is in a reservoir, fertigation water. You program it to fill the bench for a certain time. It holds the water for a certain time, and then it drains the water back to the reservoir so it can be reused. And then it just allows the plants to sit in the fertigation water and saturate with it for nutrients. And then finally, there should be vents for airflow. So I've got an arrow pointing to that, and you can see the diagram to the right of it. There needs to be horizontal airflow in this room to distribute the temperature and prevent mold and fungal growth. And so there's about a 200 micromole light intensity. We've got a 16-hour day, 8-hour dark period. So plant emergence, it's dark. Or I'm sorry, pre-plant emergence, it's dark until plant emergence. And then after that, we turn lights on for a 16-hour day, 75 to 85 degree Fahrenheit temp, 75 to 80% humidity, and 0 to 200 part per million vegetative fertilizer solution. So for the vegetative stage, largely plants gain leaves and side branches, and then they increase in height. So the vegetative stage is defined from that, I'm counting it from when the cotyledons unfold to the stem is 90% of its total length. So for our example varieties, El Dorado is 5 weeks in vegetative and White Widow is 4. And then one of the major events that happens is transplant, usually occurring 14 to 21 days after planting. And then there's two different types. I've shown them here. There's a one transplant type that you transplant it into the final pot. It'll remain in for the rest of its life cycle. And then there's a multi-transplant type based on growth. So for example, every 12 inches in growth, you would switch transplant pots. Okay. And then I just have a week by week of the vegetative stage, just kind of what it looks like. So week 1, you're getting the beginning of true leaves. Week 2, you're gaining leaves, gaining side branches. And then you continue to gain leaves, gain side branches, gain height week by week up to week 5. Okay. So what's important about the vegetative growth stage is that cannabis is one of the biggest light users out of any greenhouse crop. So I suggested a greenhouse to combine natural light with supplemental lighting to save on cost. However, if you have a greenhouse and you have clear sidewalls, then people can see your plants. So it's important to have a security system in cannabis anyway, but especially if you have a greenhouse and people know what you're growing, so you don't get theft of your plants or break-ins and things. So we have a greenhouse. Again, it's automated with an environmental control system, an irrigation control system. You can put in ebb flood benches in there as well and water pots from the bottom. It's got the same day length, higher light intensity up to 1500 micromoles this time, lower humidity, lower temp, and then it's got an increased nutrient rate. It's 1,000 parts per million, which makes sense because the plants are getting larger and larger. You need higher light, higher nutrients. Okay, so now we're in the reproductive stage. So after 4 to 5 weeks, our example varieties are starting the reproductive or flowering stage, and this is characterized by developing flowers. So one of the most important things during this phase is light treatment to induce flowering. So basically a short day to stimulate flowering. So this would occur at the very beginning of the reproductive phase, and lights, which we had on a 16-hour day, 8-hour night, are then switched from a 12-hour day, 12-hour night for about 5 days to induce flowering, and then they're switched back to the long days. And then the other major event is during early floral initiation, you will start to see pre-flowers identifying male and female plants. And as I said, you want the males to be removed because you don't want them fertilizing the females, because then you'll get seedy female buds, and that's considered a bad product. So you want to identify the males early in the flowering stage and remove them. So in the example, our reproductive stage for El Dorado lasts about 6 weeks and 7 weeks in White Widow. So I just have a week by week pictures of the productive stage. So weeks 5 and 6, you've got the flowers just starting to form. It's kind of what they look like. You can identify the pre-flowers, which are in the leaf, the axils there, and remove the males. And then your flowers get larger, you get more of them. They get even larger, you get more flowers. And then at the end of flowering, the buds start to mature and the trichomes start to mature. And I'll mention what trichomes are in a sec. So the reproductive growth space is basically the same exact space as the vegetative space. It would be a greenhouse because you need really high light levels for both the vegetative and reproductive stages. The major difference between these two is that before you were using a vegetative fertilizer, which was higher in nitrogen, lower in phosphorus and potassium. And now that you've switched to reproductive, there's a bloom-oriented fertilizer, which is lower in nitrogen, higher in phosphorus, potassium. It's supposed to increase cannabinoid quality, but basically it's like tomatoes. If you give it too much nitrogen during the reproductive phase, you don't get appropriate fruit development. So you want really good flower development, so you switch fertilizers. Everything else is pretty much the same as far as conditions for the greenhouse. Okay, finally, we come to the harvest and post-harvest stages. And so in both of our example varieties, they finished up their reproductive at the end of week 10, and then we can harvest week 11. Dry down is usually 1 to 2 weeks post harvest. So we'd finish dry down on week 12, curing week 13 to 15. So it usually lasts 1 to 3 weeks post-harvest and occurs 3 to 5 weeks post-harvest. And then finally, we have storage, or you can sell the product on week 16. So the entire cycle time is about 16 weeks for both of these varieties. And then they can either be stored or sold. Okay. So as I said, I mentioned trichomes. So trichomes are like hair-like structures on the female buds, and they actually secrete the resin containing the active cannabinoids, so THC and CBD. When determining harvest, growers use a magnifying scope to observe the trichome color. So the clear you see on the left, that would be considered too early to harvest. It's not considered potent enough, but you can harvest either when the trichomes are cloudy white or an amber. And then there are subphases of harvest, like half clear, half cloudy, half amber, half cloudy, that you can harvest in, that are supposed to have different effects on the user, and they kind of relate to that chart I showed. Are they more sedative? Are they more energizing? They say it depends on when you harvest. There are 2 methods of harvest. There's wet trim and dry trim. Wet trimming is removing branches and then removing buds and placing them on trays. So I have a picture of that on the left. And then dry trimming is cutting an entire branch and hanging it up. So I have a picture of that on the right. So basically, once branches are dry, the buds are removed pre-curing. So they're essentially really similar processes. Wet trimming has 2 steps and dry trimming has 1 essentially, but you want to end up with buds for curing, just buds, no branches. You want to let them dry to 14 to 15% moisture pre-curing. And the dry down space is just like a vegetative space, but no irrigation, no conditions needed for plants, but it's basically a walk-in growth chamber. It needs venting to allow for airflow, and it really, really needs to be sterile because mold or fungal diseases can wipe out an entire crop if your dry down space does not have adequate airflow or is not clean before you put plants in there. So a lot of these have antimicrobial filters on the air vents. They have UV light treatments. They have removable wall panels. And this is basically a room for storing racks to let things dry for curing. And so you keep humidity at about 50 to 60%, and the temp is a little bit lower at 64 to 68 degrees. The curing is after dry down, and you can actually use the exact same room. And if you have the wet trim process, which is where you end up with the buds on the racks, you can just leave it in the same room and let it cure. It's allowing more moisture to leave those buds to supposedly increase the cannabinoid content and increase the potency. It's at the same temperature as the dry down space. It needs to be sterile because, again, mold or fungal disease can totally wipe out your crop if you don't have a sterile environment, and it has to have airflow. Some different curing methods are just leaving it on racks in a room like this. There's also freeze drying, which a lot of big producers use, and I have a picture of industrial freeze dryers on the left there. And then for smaller operations, they'll use jars, but there are companies that develop desiccant packets to get the curing moisture, get to the correct curing moisture. Okay. And then the last step is storage, and that was about week 17 or week 16 on our example varieties. And this is just, you have long-term storage or short-term storage. Long-term storage is 6 months to 4 years. Short-term storage is up to 6 months. But important things about the storage is that the longer you store cannabis, the less potent it gets, the more it loses THC and CBD content. So it's important to not store past 4 years, or if you don't have to store, don't, because you'll make it less potent. And basically, storage spaces need to be free of light, heat, and air to, again, prevent mold and fungi so you don't lose your crop. And short-term storage has slightly higher temperatures, about 64 to 68 Fahrenheit. Long-term storage is about 14 Fahrenheit. And often people will use freezers. And then secondary containers within the freezer. So I have a picture of a couple of those. The center one is just a plastic container that would go in a freezer, because that picture on the right is not an ideal storage method, because the buds get really brittle, and if they're stacked like that, they'll break. So you really want like a secondary container, and a lot of people use vacuum sealing, and they don't stack them very high so the buds don't become basically a little, crumble up. And so that's the entire process of a cannabis workflow. So I wanted to say thank you to my advisor and committee for their help with this project, and ask if anybody has any questions.
I think we have time for one or possibly two questions.
Did you have any problems with diseases or insects in the greenhouse?
So I had to plan a workflow. I haven't actually tested it. So really the next phase would be determining if what I'm saying about collecting plant metrics and trying it out on a tester variety would actually work in a greenhouse. But I planned essentially a workflow for cannabis to test out different varieties and grow them.
So I understand using seeds for hemp, but my understanding is most cannabis production is from clonal varieties. So why start with seeds for this project?
It just depends on what type. It's just the purpose of what you want to do with it. So in clonal varieties, you're cloning, or I'm sorry, clonal production systems, you're cloning the mother plant. And so you just have copies of that mother plant. You don't have any males. So if you want to breed something and have sexual recombination, you need male plants. So you end up with female plants if you just do cloning. So if you want to do large scale production really quickly, you can do a cloning operation. But if you want to develop traits, you can do a a seeding operation, or maintain an inbred line is also what they use it for.
So if I can chime in one point on that, Megan, for Dr. Murray's benefit, he's obviously a churchgoer. Type 1 marijuana a year ago was $2,000 a dried pound of flower. Now it's $200 or $300. So a lot of these people that are used to spending a lot of money on vegetative propagation are going to have to adjust their methods. So we asked Megan to approach it this way, assuming they're going to have to do that in the near future.
Thank you.
Let's thank our speakers again for a good job.